Safety in Microbiology Laboratory by Nakanakanahiwa Wingfield. A microbiology laboratory is a unique environment that requires spatial practices and containment facility in order to properly protect persons working with microorganisms. Safety risks that are associated with microbiology laboratory includes chemical, fire, electrical, radioactive, and the most important is biohazard. Biohazards are the biological substances that may present a health risk to humans. Clinical specimens are potential biohazards to laboratory personnel, so you need to be aware all the time when you need to handle these kind of specimens. Possible routes of infection in microbiology laboratory. The first route is airborne. Some of the microbes can be airborne, for example, fungal spore and virus. Ingestion. Sometimes you can be accidentally ingest or intake the culture spill. So you will need a proper protection. Direct inoculation is by injecting some of the contaminated culture into your body. And the last one is the mucous membrane contact by contacting all those culture on your skin. Here are safety guidelines for microbiology laboratory. When you need to work in the microbiology laboratory, there are so many rules and regulations that you need to consider before working in the lab, because whenever you make an accident or something happen, it can be very fatal. So here are some do's and don'ts guidelines. First, laboratory procedures must be read prior to attending lab session. Hand washing is required before arriving and leaving the lab. No food or drink is permitted. Cosmetics are not to be applied. The use of laboratory gowns, clothes is required. Do not wear gowns, clothes to other non-lab areas. Wear appropriate shoes, and shoes should cover the entire foot. Disinfect work areas before and after use with 70% ethanol. Keep your workspace free of all unnecessary materials. Mouth pipetting is prohibited. Label everything clearly. Do not pour anything down the sink. Replace caps on reagents, solution bottles, and microbial cultures. Inoculating loops and needles should be flame sterilized. Turn off Bunsen burners when not in use. Treat all microorganisms as potential pathogens. Do not take cultures out of the laboratory. Dispose of all solid waste material in a biohazard bag. Glassware, sharp objects should be discarded in the provided containers. And here are some safety guidelines when you make an accident by doing the spill, even though it's a minor or major spill. If you make a minor spill, please report spills, accidents, and any or injuries immediately to your instructors. And then clean small spills with care. And whenever you make a large spill, please seek help for large spills. And here are some brief guidelines for cleaning up small spills. First, you will need to contact your instructor. Then soak paper towels in 70% ethanol, and then place the towel around and on the spill area. Then allow 10 minutes contact time. After 10 minutes After the 10 minutes contact time, you will need to clean the spill area with fresh towels soaked in the disinfectant, working from the outer edges into the center. Then place the paper towels and gloves into a biohazard bag. And importantly, wash your hands with a disinfectant soap. For working in the microbiology laboratory, you always need to consider that all of the material are contaminated. But after working, we would have to eliminate or dispose of all the biohazard waste by using these 3 ways. The first way is sterilization, the second disinfection, and the third is antiseptic. Sterilization is the process that destroys all microbes and spores, which includes autoclave and dry heat. Disinfection is the process that destroys or irreversibly inactivate microbes, but not the spore. These include 70% alcohol, chlorine compounds, sodium hypochlorite. And the third one is antiseptic, which is the process that uses the chemical germicide for use on skin or tissue. The thing that we can use is 70% alcohol. Here are some questions that I would like you to recall what you have learned from the safety guideline from microbiology lab. The first question: an infection that may occur as a result of accidental needle sticks or through broken glass is classified as which of the following routes: airborne, ingestion, direct inoculation, or mucous membrane contact? Second question: autoclave standards for decontamination of most biological materials are: first, 100 degrees Celsius for 45 minutes. Second, 121 degrees Celsius for 45 minutes. C, 121 degrees Celsius for 60 minutes. And D, 100 degrees Celsius for 40 minutes. The last question: which germicide is intended to destroy all microorganisms and their spores on inanimate surfaces? A, sterilizer, B, disinfectant, C, antiseptic, and D, antibiotic. Guess the words. The first word is the biological substances that may present a health risk to humans. Second word, disinfectant for work areas. Third word, samples from patients. Find the words. There are 5 words that are hidden in here. Could you please help me find them? Thank you.